class, so I'm going to actually be joining you in video in this one. So, hello. We are talking about chapter 7. This is membrane structure and function. So this whole chapter we are talking about what the membrane is made of and how the membrane works. It does some important things like protecting things that are inside, protecting from things that are outside, controls what can move across the membrane, and in a couple of chapters, we'll learn how the membrane even is involved in making energy from food. So the plasma membrane is the boundary that separates the living cell from its surroundings. In this picture, this red and gray is the water outside of the cell and the water inside of the cell. So these yellow heads are the phospholipid heads, and these green tails are the hydrophilic tails on it. And then here's the other side, because your plasma membrane is a bilayer. Here's the other phospholipid heads and tails. The plasma membrane has this really important quality called selective permeability. That means that it is permeable. It lets some things pass, but it is only selectively permeable. It gets to select what it is permeable through. So it lets some substances cross it. Some can cross it easily. Some can cross it a little bit less easily, and some aren't allowed to cross the membrane at all. Often a lot of what controls what can move in and out of the cell are going to be transport proteins. In this picture, this is a picture of an aquaporin. That's a protein that transports water. So you can see these blue alpha helices. They're just rolled up. They're making just a little channel in the membrane here. And you can see these little water molecules are able to come through. If you were to look at a lot of pores or channels from the top, you would see it forms, a barrel-like shape that it has a little hole in the middle, a little tunnel. In this case, this is a potassium ion channel. So potassium ions can fit through. If it's bigger than a potassium ion, it's not going to fit, so it won't be able to fit through there or move through there. Some channels are going to be selective for they need to have a certain charge or they need to have a certain size to be able to go through there. So let's watch this video about cell membranes. See how they are moving around. They're not totally still. These phospholipids are able to move. And when those phospholipids move, it also allows proteins to move. Because sometimes those protein channels need to be able to shift a little bit to open up or close back up. Some of them are just going to be sitting there just always open, but some of them need to be able to shift and move. Some channel proteins are going to need to interact with each other, move closer to things or move away from things. So that allows for things to move around and shift as the phospholipids are moving also. So phospholipids are the most abundant lipid in the plasma membrane. So we talked about that phospholipid bilayer. So here's a layer and here's a layer. They each have hydrophilic heads and a hydrophobic tail. Here's the other layer, hydrophilic head and hydrophobic tail. If you were to zoom in on these two phospholipids, you'd see this. So the hydrophilic head out in the water outside the cell, hydrophilic tails interacting with each other, and then hydrophilic head interacting with water inside the cell. So these phospholipids are called amphipathic. Amphipathic, so if you think of amphi like an amphibian, amphibians can live in water and out. They have two ways of living. So amphipathic molecules have two different distinct parts. They have a hydrophobic region and a hydrophilic region. So the hydrophobic regions, hydro means water and phobic means fearing, so they don't interact with water. That's these tails. Hydrophilic, hydro means water and philic means loving, so these hydrophilic heads interact with the molecules inside and outside of the cell. So the hydrophobic tails are sheltered in the middle of the membrane, and the hydrophilic heads are exposed to the water inside or outside of the cell. We just saw that video that showed that the phospholipids are moving around and the proteins are kind of bobbing around and inside of it with it. That is what the fluid mosaic model describes. You have a fluid membrane, but it's not just phospholipids. It's a mosaic. It's made of multiple different pieces, all working together to create the final thing. So you've got the phospholipids, but you've also got proteins. And proteins are going to be very specifically put in different places. You've got carbohydrates. Carbohydrates would be like sugars. We have glycoproteins. This would be a sugar attached to a protein. And glycolipids, which would be sugars attached to lipids. And that whole thing is moving around, functioning together, and that's what makes the fluid mosaic model. That's what the fluid mosaic model describes. Membranes are held together mainly by weak hydrophobic interactions. So these interactions between the hydrophobic tails of the phospholipids on both sides of the membranes. Lipids and proteins can move sideways around, so they can kind of shift a little bit here or there. Every now and then you might see a phospholipid flip from one side to the other, but that is very rare. That doesn't often happen. Mostly it's going to be this side phospholipids moving around, and then these sides moving around on their side. So one thing to remember, when things get cold, they freeze. Cell membranes are no different to that. If that cell is getting in an increasingly cold environment, that cell membrane could switch from that fluid mosaic model where it's moving and functional to it could freeze into a solid state. If it is frozen, it's not shifting around, it's not going to be very functional, it's not going to keep that cell alive for very long. So cells are going to do things to protect from freezing. One of the things they can do is change the types of lipids they have in them. So if you have a lot of unsaturated fatty acids, remember that unsaturated means that you have a double bond. You've lost some hydrogens and created a double bond, and that double bond forms a kink. So it just is going to kink. Those double bonds of an unsaturated fatty acid are going to spread the molecules out, and that helps it not freeze. So an organism that lives in a very cold environment may have more unsaturated tails, and an organism that lives in a more hot environment may have more saturated tails in order to keep that viscosity in it. Cell membranes, especially also animals, so our cells, we have a cholesterol in our cell membranes. Cholesterol will keep your cell membranes fluid by stopping those phospholipids from packing too tightly together. So it just kind of does the same thing as what we saw with unsaturated tails. It just gives a little bit of a kink. So that will keep your membranes fluid. The membranes have to be fluid. They're going to be about the consistency of like olive oil, and if they're any more solidified than that, they don't function as well. So that's why some animals or plants or some species may have unsaturated fatty acids in their cell membranes, and some may have cholesterol. So here, this is kind of what we just talked about. At warm temperatures, cholesterol stops phospholipids from moving too much. At cool temperatures, it stops them from freezing tightly together. Plants often have cholesterol in them, but they don't use cholesterol in their cell membranes. These are different steroid lipid, but they still have some sort of lipid to keep their membranes fluid. So, you do need some cholesterol in your life. Your body can make your own cholesterol. You don't have to be eating a bunch of cholesterol. But cholesterol isn't always a bad thing in your body. Environmental conditions can affect, so, different organisms in different environmental conditions may have things, different compositions of saturated and unsaturated tails. And being able to change the lipid compositions in response to a temperature change has allowed for life to evolve on Earth in more extreme conditions. So organisms were able to survive colder temperatures by doing this, and to evolve in warmer temperatures by doing this. So that word collage paints a picture for me of something like, or the word the word mosaic paints a picture in my mind of something like this. So it's a bunch of different pieces of tile all working together to make one big, beautiful picture of a bird. So like that, a membrane is also a collage of different proteins, different phospholipids, different lipids. Often proteins are going to be clustered together in groups if they're functioning together, and embedded in that fluid matrix of the lipid bilayer. Phospholipids, though, are still the main backbone, that they provide the scaffolding for the plasma membrane. But proteins are what is going to determine most of what the membranes are doing, most of the functions, because that is what proteins do. They do things. There's two different types of proteins, peripheral proteins and integral proteins. Peripheral proteins are just bound to one surface. They're on the periphery. Integral proteins are integrated. They run through the whole thing. So here you see a protein that crosses both membranes. You can see it on the outside and on the intracellular side of this cell membrane. Is this peripheral or integral? It penetrates the whole cell, so it is an integral protein. Integral proteins that span the membrane are called transmembrane proteins, and they will have hydrophobic regions here that are going to be nonpolar amino acids, and they'll have hydrophilic regions that are going to be more polar or ionic amino acids. So they're able to coil up and form this shape that allows it to penetrate both the hydrophobic and hydrophilic regions of the cell. A lot of proteins that go through the cell will span the cell membrane 7 times. They're called integrins. And so you can see here's 1, 2, where does it go? 3, 4, 5, 6, 7, something like that. So you can see 1, 2, 3, 4, 5, 6, 7. Those are integrin proteins that will span 7 times. They go up and down through the membrane. That's not a test question, but that's what I did a lot of my graduate school work on, so that always interests me. So cell surface membrane proteins can carry out several different functions. One is transport. So here it just forms a channel, lets anything that fits goes through. Here it forms a very specific type of receptor channel where you have to fit in it, and then it'll open and let it out. Some membrane proteins are enzymes. They're going to be changing things down. Especially in a lot of prokaryotic cells, this is where a lot of metabolism occurs, where sugar gets broken down to energy. Signal transduction. So in something like people, we need to send signals from one cell type to another. So in our cell membranes, we have these receptors that, your your body sends a hormone or some signal, and then your cell receives that hormone and then can send this message on, and inside this protein will go cause some change inside the cell. One is cell-to-cell recognition. So, your immune system is going through your body looking to see, is this my cell or is this an invader? If it's an invader, I'm going to kill it. So your white blood cells need to be able to recognize, this is my cell, protect it, or this is not my body, kill it. Intercellular joining. It should be something like this. 2 cells need to connect together and stay closely joined together so that they can function together in a tissue or in an organ. And then there are membrane proteins that hold this membrane attached to the cytoskeleton and to the extracellular matrix. So that cytoskeleton that's inside the cell and the extracellular matrix that's outside of the cell, that membrane protein connects through to all of these. All of these that we're looking at here are all transmembrane proteins, except here, this one is not an integral protein. That's a peripheral protein. It's on one side. This is on one side. It's a peripheral protein. And this is only on one side. It's a peripheral protein. So that's the difference between an integral protein and a peripheral protein. Please be able to remember those terms separately also. Cell surface proteins can be important in the medical field. So HIV, this is a virus that can go on to cause an infection called AIDS. I hope you've heard of this before. This is a pandemic across the world right now. So HIV, to be able to infect your cell, first it's going to bind to your cell. And the way it binds to your cell is through a receptor called CD4 and a co-receptor called CCR5. Some people, their bodies do not make CCR5. They only have CD4 on it. And without CCR5, HIV can't firmly bind to the cell. So it won't get in. Here, a cell that has both CD4 and CCR5, the HIV gets into the cell. But with only CD4, the cell doesn't get into the cell. So there's a lot of drugs that are now being created to block this co-receptor. So your cell acts like this. It stops HIV from binding to CCR5 so HIV can't penetrate and get into the cell. So things like prophylactic, like PrEP type of drugs that stop people at high risk who may be exposed to HIV. It can protect them from actually getting infected with the virus because the virus can't bind to their cell. So that's one important medical implication of receptors on your cells. Almost all viruses are going to bind to some receptor. So they do some sort of something similar to this, binding to some receptor on your cell. Cells can recognize each other by binding to molecules on each other. Often they're going to be binding to carbohydrates or to glycoproteins, sugars attached to proteins on the extracellular surface of the plasma membrane. If a carbohydrate is bonded to a lipid, it's called a glycolipid. If it's bonded to a protein, it's called a glycoprotein. So glyco, sugar, lipid, lipid. So it's a sugar and a lipid together. Glycoprotein, a sugar and a protein attached together. Sugars on the extracellular side of the plasma membrane are going to be different between different species. They're going to be different between different individuals, and they're going to be different even in different cell types of one person. Because they can vary in individuals, this is why you want to match people, like when you're seeing someone's getting an organ transplant, that someone is a match or not. It means that their carbohydrates are the same. So if their carbohydrates are the same, their body would recognize it as an acceptable part of their body and allow it to function. And if the carbohydrates were different, their immune system might say, this is different from me, I better kill it. So matching those carbohydrates on an organ that's being transplanted is very important. And also, sometimes we get organs or something from other species that also could be important there. A cell is also going to need to be able to exchange materials with its surroundings. So sometimes it's going to need to take in food or take in some sort of molecule for an energy. It's going to need to take in some sort of molecules to be able to turn it into the things that it needs to grow. It's going to need to be able to get rid of waste. It's going to be able to send things out. So a cell has to be constantly exchanging what's inside of the cell and outside of the cell. But it doesn't want to just let anything go in or out, because it needs to protect some things that are in it, need to stay in it to function, and some things that are outside of it could harm the cell, and you don't want to just allow anything to come in. So plasma membranes are selectively permeable. Things that need to go out will be allowed out, but things that shouldn't go out will be kept in. And the same thing, things that it needs to come in will be allowed in, but things that it doesn't want to come in won't be allowed in. That is what it means to be selectively permeable. So small hydrophobic or nonpolar molecules, so things like carbon dioxide, small hydrocarbons, can just go right through that lipid bilayer, and they can pass through the membrane rapidly. So it doesn't need any help, it just goes straight through the cell membrane. Things that are hydrophilic, things that are ionic or polar, they're not going to cross the membrane so easily because they're not going to be able to interact with that hydrophobic core of the bilayer. So instead of being able to just go straight through the membrane, it's going to go through protein channels. So those protein channels in the membrane are going to be key to regulating what is going in and out. If there is a channel to allow something in or out, it can go in or out. If there's not a channel, it can't go in or out. Transport proteins will allow passage of specific hydrophilic substances across the membrane. Some transport proteins are called channel proteins because they're going to form a channel that certain molecules or ions can use as a tunnel. One channel protein we looked at the very beginning of this lecture was aquaporin. Aquaporin allows water to pass through in and out of the cell. Some types of transport proteins are called carrier proteins, and they're going to be specific for one type of molecule, and when it binds to that molecule, it's going to change shape, and that change in shape is going to allow them to cross the membrane. So channel proteins are just anything that fits can go through, but these carrier proteins are going to be specific for what it is moving through. But both types of this is not going to take energy. It is going to allow things to come in. If there is more outside than inside, it will pass through. If there's more inside than outside, it'll pass out of the cell without any energy. It always is only going to move from high concentration to low concentration. So if you already have a lot of it inside of your cell, this is not going to allow you to hoard more than you have in your environment. If you are needing to bring it against that concentration, you're going to have to use energy, and that's called going against the concentration gradient if you're building it up where you already have more. And that is going to be an entirely different type of transport that we'll talk about later in this chapter. So diffusion is the ability of things to just kind of spread out evenly, right? Like if I poured out my drink, it's not going to stay in a pile where I pour it, it's going to spread out. It's going to diffuse. If I, put a drop of food coloring in a bowl of water, that drop is not going to stay there. It's going to spread out. That's what diffusion is. So here, we have a pseudomembrane, right? A little barrier pretending to be a membrane, and you put a bunch of dye on one side of it. It's going to cross back and forth here. So at the beginning, it's going to only go into the cell. As you start building some in, you'll have some go in and some go out, but mostly it's moving in. And then eventually you'll hit equilibrium, where you have some moving in and some moving out, but they balance each other, so you're at equilibrium. It's equal on both sides of that membrane. If you had two different types of dyes, so if you put orange on one side and purple on one side, they're not just going to say, oh, there's already dye there, I'm fine, I don't need to move. They're each going to move independently. So the orange is going to spread itself out until it is balanced on both sides, and the purple will spread itself out until it is balanced on both sides. So each type of molecule finds its own equilibrium independently of the other types of molecules that are there. At a dynamic equilibrium, you can still have many things passing through the membrane. So you can see orange is still going in and out, purple is still going in and out, but they're at a balance. So you keep a consistent concentration on both sides. Here's a video about diffusion. Ooh, let's pause that, back it up, see if we can get some sound on here. Maybe that'll do it.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Okay, so diffusion is just things moving straight through the membrane, doesn't need any help, but it only moves until it gets to equilibrium. Okay, it doesn't take energy. That's passive transport. That's all what diffusion is. So here's a video about membrane selectivity. Oh goodness, I'm going to have to do this on every video, aren't I? Okay.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Okay. So, passive transport is letting something move across the membrane without using energy. So substances are moving down their concentration gradient. They're moving from where they're very concentrated to where they're less concentrated until they find that region of equilibrium. No work has to be done. Just like you don't have to, if you drop if I poured out my water, I wouldn't have to do work to get it to spread out. It just does that. That is just what chemicals do. And so this is called passive transport because it doesn't take energy. Cells just make it happen. Let's pick up with osmosis in part 2. Before you watch that, maybe watch the Amoeba Sisters osmosis video. This tends to be the section of the chapter that gets people tripped up the most. So watch that Amoeba Sisters video, then come back and watch part 2 of our chapter lecture. I'll see you in a little bit.